Well, ladies and gentlemen, it is about time to start our third session, our third panel session. Welcome to our afternoon session on biomedical sciences. We're going to start introducing the participants. Mr. Erwin Neher, Nobel Prize laureate, medical sciences, specialist. As you all know,
there's so many so-called incurable diseases. And Dr. Neher, empezaremos por hacer una pregunta con la
I'm going to start asking a question. The scientist will introduce himself. I'd like each of you to introduce briefly.
In that question that he's been working, you received the Nobel Prize for discovering ion channel currents. The Nobel Committee expressed the expectation that this discovery will lead to progress in biomedicine. Did this happen?
Well, let me, in terms of introduction of myself, first say that I'm a physicist by training, working in Germany, in Göttingen, at the Max Planck Institute, nowadays on synaptic transmission. That's a problem of how nerve cells communicate with each other. As you said, I received, together with Bert Sackmann, a colleague of mine, the Nobel Prize for discovering ion channels. And your question was whether the expectation of the Nobel Committee that this would eventually lead to benefits for biomedicine, whether this came true. There are several aspects to this. Ion channels are pores in the membranes of our cells which mediate the most diverse functions, signaling functions and other functions. When we began studying these channels in around 1972, we just wanted to find out whether one could, according to the concept of channels, which are these pores which open and close in response to certain stimuli, whether one could record electrically current changes, discrete changes in current, which signify the opening and closing of these channels. When we were interested in that because we knew from the earlier work of Hodgkin-Huxley in the 1950s that the nerve impulse was a cause of permeability changes of nerve membrane. Hodgkin-Huxley showed that when a nerve action potential arises, the membrane first becomes permeable to sodium ions. The sodium ions rush in, depolarize the cell, and then potassium. Then it becomes permeable to potassium. Potassium rushes out, the membrane returns to the basal level, and this is the action potential. We were interested to show that this actually happens by means of these channels, pores, which open and close. Now, by refining the techniques of recording currents from cells, inventing the so-called patch clamp techniques, we were able to show that indeed one could record these discrete current changes. And so we were, of course, happy to have shown that the channel concept is correct, that there are indeed these little pores operating in nerve membrane and also in muscle membrane. At that time, this was all we wanted to know. We didn't think much further. And indeed, the expectation, which then 15 years later was expressed by the Nobel Committee, that this would be good for understanding several health issues, came about through several surprises. The first surprise was that such channels, which open and close, are not only operating in so-called excitable tissues, which are nerve membranes, muscle membranes, certain neuroendocrine cells, but basically all types of cells in our body are harboring most diverse types of ion channels. By now, we know that about 200 genes encode for ion channels and other transporters, and that these molecules, which are distributed in a specific way over the different tissues in our bodies, subserve the most diverse function, such as nerve action potential, fluid transport in the kidneys, certain transport processes in the liver. Even red blood cells have ion channels which serve to keep their volume constant, and even plant cells have ion channels in their stomata to control the opening and closing of the or the excess of CO2 and oxygen to the plant cell. So this to us was a surprise, of course a very lucky surprise. The other surprise was that ion channels, since they subserve control functions, are prime drug targets. You know? So many of the drugs which have been in use over decades, their action is now understood by the influence they exert on ion channels. So, for instance, the so-called calcium antagonists, which are used for treating high blood pressure, act on special type of calcium channels by changing their willingness to open, so to speak. So, ion channel research has become a very hot topic in pharmaceutical research. The third surprise was that indeed many hereditary diseases are caused by defects in channels, in the sense that different kinds of channels don't operate in their usual way anymore. So in this sense, our initial discovery, or the tools we provided to study ion channels, indeed had major influence on biomedicine, but nevertheless, even now, 25 years after we established these tools, it is hard to identify, say, specific drugs for which one could say that they are a direct consequence of ion channel research. Although many types of the working of many types of drugs is now being better understood by these possibilities, still I think it takes another 5 or 10 years until the full potential becomes noticeable in the real world, in the clinics.
Thanks a lot. Then we got 10 years. Tenemos 10 años para esperar.
About 10 years ahead to see those new pathways of ion channels. And it's a very important prize you have got. Now, Dr. Carballo, who's a specialist, also worldwide expert, and he prefers English instead of Spanish because that's what he has suggested. And Dr. Carballo is going to start his presentation with a short description on where we are now in the field of AIDS. And is
Dr. Carballo, all right.
Thank you. I remember that a few years ago, sitting not here but in the campus in the Canarias, sitting with Luc Montagnier, who was one of the people to identify the HIV virus, listening to him say how pessimistic he was about the biomedical advances that would be or not be made in the area of AIDS, and how much he expected the more social and behavioral part of prevention to be successful. Looking back on the years that have passed since then, I think one of the things that we have realized is that in the area of prevention, of changing behavior, of reducing risk, we have been eminently unsuccessful, and that in the biomedical area, we have been eminently successful. So that in the period since, let's say, 1985, when the virus was discovered, we have been able to go on and to develop the type of antiretroviral therapy that is now making it possible for millions of people who are infected with HIV to live good quality lives, remain economically active, and so much so that last year, Professor Herschel in Geneva, the professor of infectious diseases at the Geneva Hospital, publicly stated that discordant couples, that is to say, couples in which one partner is infected, but who is on antiretroviral therapy, that couple need no longer use barrier methods, condoms, to protect themselves against HIV. A relatively controversial statement to make, but nevertheless indicative of the extent to which we now have a product that is able to reduce the level of viremia in an infected person and hopefully cut down the cycle of infection, not only from one person to another, but within a given population. At the same time, I think that these advances in the biomedical aspects of HIV and AIDS have introduced a series of paradoxes that we were entirely unprepared for. For example, of the 33 million people who today live with HIV, the infection, and who are progressing to a condition that we normally would call a condition of HIV-related diseases, AIDS, only 4 million people today have access to the therapy which is life-saving. That is 1 million more than last year. And we hope that next year we will be able to say that another million have access to this therapy. But even if we do, it means that something like 25, 26, 27 million people still do not have access to a life-saving drug. And I think that the issue of antiretroviral therapy in the context of AIDS has introduced new questions in the area of public health. Who has the right to access to care? What are the characteristics of those people, be it in terms of political status, geographical status, socioeconomic status, that will make them eligible for life-saving treatment? I think it has also introduced a number of difficult questions concerning the pricing of drugs and the relative pricing between so-called developed and so-called developing countries. So I think that as we look at the history of HIV/AIDS over the last 15 years, on the one hand, we can say there's been tremendous progress beyond our expectations, and at the same time, we are confronted with a disease which is still growing. 7 million people are newly infected with HIV every year. And another dilemma, and that is that if we were to look at the prevalence as opposed to the incidence of HIV, the more we are able to make antiretroviral therapy available, the higher the prevalence of HIV automatically becomes, with all sorts of cost implications, with implications in terms of the types of services that are required to make antiretroviral therapy technically viable in a correct and efficient fashion.
Thank you, Dr. Carballo.
Thank you, Dr. Carballo.
We are not short of them. They are high quality. He came all the way from Israel.
Yes.
Let me ask you a question. Yes. About de pronto, we say in Spanish. Yes. Why Israel has given so many and so good scientists, basic research scientists?
I hope you are right.
I am right, I know.
We have some good scientists, and why? Well, people are interested in science in Israel. Can you hear me with that contraption? Yeah. But I wanted really not to talk about my country, but even though I like it very much, I wanted to talk about science. And
please don't. Then I will ask you on. I know you were working in basic research in proteins, as far as I remember.
Yes.
And I would like you to give the latest news on precisely on that, on your field.
Okay, well, the latest news. Well, I'll start with some old news, and then I'll go to the latest news. So I would like to actually to follow what the previous two were saying, because what you can see is how in science you cannot really know what would be the future consequences of what you are doing.
Yes.
You have to do good science, and then you don't worry about anything else. He wanted to know how channels are working, and now we have drugs. In HIV, people just wanted to know how the virus multiplies, and they found that there are a protease that is needed for the virus to multiply. Once you knew about the protease, and you identified the protease, then you could also devise new drugs. And then you have drugs, and then you have an acute illness converted to a chronic treatable illness. Of course, you have the public health issues that you mentioned. That we cannot solve. That's only the society can solve. But in science, I think your story and also mine shows how we should not worry about applied science and basic science. We should do good science. If you do good science, eventually it will help mankind. So for example, I am interested for many years in how proteins in our body are broken down to their building blocks, amino acids. And I was also interested just because I was curious. I noted when I was very young, postdoctoral fellow, I noted that protein degradation in our cells needs energy, which is not the case with protein degradation outside the cells. Outside the cells, it doesn't need energy. Actually, it liberates energy. But in our cells, it needs energy. And I was asking why. And the question was, well, maybe because it's very selective. But how does it happen? And we found out. It took, by we, I mean a whole group, and which included also Ciechanover, who was then my PhD student, and he should have been here, but he's sick here with another virus, H1N1, I guess, or one of these. Anyhow, and with Erwin also, we found out that proteins are energies needed to target, to mark a protein to be degraded by being linked to another protein called ubiquitin. And then we worked on what this system does. It turned out to be very important in almost every processes, because the proteins are the machines of our body. So it's important in everything that the cell does, including cell division, including getting rid of bad proteins. So it was just curiosity-driven science. But then it became applied, because cell division is related to cancer. When in cancer, cells divide when they should not divide. So cell division gets out of control. So suddenly, this basic knowledge that we learn by just being curious, by just trying to find out how things work and how our cells work, now it became relevant to cancer. And now there are drugs that are based on our discoveries that are good against certain types of cancer. There is a good drug that is effective against a certain cancer, bone marrow cancer called multiple myeloma, which is actually targeted against the ubiquitin system. And it is a very valuable drug for this disease, which makes it a disease which was rapidly rapidly fatal to be a manageable disease. So there, I think to give a couple of more years of good quality life to a person or to many persons, that's an achievement. So I think that we should not, maybe the take-home lesson from this story is that we should not be worried about applied research or basic research. We should do good research. If you are doing good research, eventually it will become good to humanity. Now, what is the newest? The newest thing, I'm now studying how this system is involved in the accurate division of chromosomes. When our cells divide, then each new cell has to get exactly the same number of chromosomes. Otherwise, the cell would go into chaos. You would have diseases, you would have cancer. So it turns out the ubiquitin system is involved in that too, in that accuracy of the chromosome segregation. So that's what I am doing right now.
Great, thank you. After all, life is the transformation of energy in information.
Yes.
And it's a complex process.
Yes.
That you know a lot about, sir. Tenemos a Rosario.
Rosario here with us. She speaks Spanish. We are in Madrid, after all. Rosario Monataya, researcher from the CSIC, and she's an expert in the field of Parkinson's disease and cocaine intake as a disease. Do not misunderstand me. And the lady doctor has been analyzing a subject that is a very important subject, which is vaccination against addiction to cocaine. Those are big news. And she thinks that it is a therapeutical opportunity, or is it a waste of time and money? Where are we? Would you kindly elaborate on that?
Well, I will start by saying, as everybody knows, and as you know, that cocaine is a psychostimulant, and it is one of the drugs with the highest addictive power. We know this because of two things which are obvious, and that is that cocaine consumption, or rather cocaine abstinence, is the one which produces the highest number of relapses. And this gives us an idea about the addictive power of cocaine. And also that patients are the drug addicts who seek medical help the earliest, and this is a sign of how bad they feel, that they lose the line between the pleasure they feel and reality. The line gets blurred, and then they seek medical help. Cocaine, as you know, is a psychostimulant which acts because it blocks the dopamine transporter and therefore interferes with the intake mechanisms of neurons in order to save energy and at the same time to control the neuronal or the neuron activity, and therefore it causes an increase, an incredible increase in dopamine in the synaptic cleft, and activates all that comes, the neurons that come later. Cocaine consumption, among other things, causes an increase in the motor activity and plastic changes in neurons, which do not only become more excitable, but we have also seen that the neurons of the limbic system, which is the reward system, have more dendritic spikes. So they cause long-duration, long-standing plastic changes, and they also cause changes in the limbic system and the reward system. One of the main animal models to test the addictive power of a drug is administration to animals. They are placed in boxes which have been preconditioned, and the mouse, or the rat in this case, presses a lever, and each time he does so, he receives a cocaine dose, and in this way they learn how to self-administer cocaine. This is a valid model because it is the addiction model par excellence, which represent the causes of an addiction to a specific drug, toxic drug. I think that having a vaccine is a good strategy, I believe, to try and reduce cocaine consumption. Well, the results have been published recently in the media and in the scientific literature, and they have been tested in addicts to cocaine. This had already been tried in those animals who self-administered cocaine, and we realized, it was seen that in those animals it worked perfectly well. In Yale University, the study carried out did not provide the expected results. They were not as spectacular as in the animal model that I mentioned before. Apparently, and really, it hasn't worked in the way we had expected, because in vaccination, there has been quite a low level of vaccines or antibodies in the serum, in the blood serum of those addicts. And as the number of relapses in those drug addicts, cocaine addicts, decreased substantially, or increased, substantially, didn't reach 100%. About 40% of the subjects submitted to the study, well, the total number was 115 to 120 drug addicts who submitted themselves to the study. Have those persons, and the study was very well designed because it included placebo, a placebo group compared with the drug addicts who had been vaccinated against cocaine. The vaccine consists mainly in the fact that the body will produce a molecule that may sequester cocaine each time the cocaine goes into the individual or the person's system. It prevents cocaine to go to the central nervous system, and therefore the cocaine effects are blocked. This is the vaccination mechanism against cocaine. And I think it is a good strategy in order to be able to decrease cocaine addiction. I just think that in this study, in this specific case, the proper or the right levels of antibodies were not achieved to state that it was going to work in 100% of the subjects in the study. It has only worked in 40% of the subjects, as I said before, and those in which it worked, well, they differ from the others in that their antibody levels were above 40 micrograms per milliliter blood. In those in whom it didn't work, the antibody level was quite lower, which mainly means that the strategy is good, and that if we continue, if we go on trying to obtain vaccines with a higher antibody titer, we might be on the right track, and we will be able to reach our objective. However, this immune therapy, which is because this is the name we would give to it, well, I think that it shouldn't be monotherapy. It would be much better to combine it with other therapies, such as, for example, psychological therapy, so that the individual, during the abstinence, the withdrawal period, decreases anxiety, stops having persecution mania, and it wouldn't be bad to combine them with other pharmacological therapies. But unfortunately, today we don't have any pharmacological therapy which may provide spectacular results that would allow individuals to stop taking cocaine. Thank you very much. I will continue here with mineral water, and I will continue with it. Well, I am announced that we have Dr. Esteller with us. Doctor, thank you very much. He's just had a traffic accident. We hope it is not serious. He's here, all whole. It's a real relief. Welcome, and I'm going to allow you to take a seat, and then I will ask you to speak about oncology because that is your area, your field, and I will ask you a very general question, so get ready, please. And I will take this opportunity to ask the speakers to try and find common points in order to try to put together the common points of their specific fields, trying to find crossroads in order to enrich the scientific debate. You are worldwide specialists in your field. What would you talk about with a beer in front of you? And something related to science, of course. Please let's try to find those common points. I'm sure our audience will be very thankful for that. I have Dr. José Regidor to my left. He comes from the University of Las Palmas in the Canary Islands. Welcome. He's a specialist in neurodegenerative diseases, and more specifically, he's an expert in Alzheimer's. Well, my obvious question, everybody's question when talking about Alzheimer's disease is, where is the cause? We still don't know. I would like to start by saying that about 100 years ago, Ramón y Cajal wrote that when he was studying the brain, he was persecuting neurons, the butterflies of the soul, and that perhaps one day they would disclose the secret of mental life. With regards to common points, Cajal carried out very high-quality research, and he left us an extraordinary inheritance. Professor Neher's study about the ion channels provided a magnificent vision about the connections between neurons. That is, they're sending messages to each other, and that this takes place through a very complex mechanism. There is something which was neglected for some time in the study of the nervous system, of the brain, and that is the aging process. How do we age? How does the nervous system age? It is a system which virtually does not renew itself. We can almost say, well, of course, new neurons are generated. We know that today in some parts of the brain, but most of them, mostly, we will be buried with the same amounts of neurons that we had when we were born. But how do they age? Is aging a pathological process, or is that a natural process in the system? And in this regard, what is clear to all of us is that there is no vaccine against aging, or there is no private clinic which may solve that problem. We will continue to live with the life we have, and with the years, the age, we will turn progressively. Aging started to worry us because we see that there is an increasing number of elderly people in our society. Europe is beginning to be an old continent, but within Europe, Spain, according to populational studies, very soon, in the year 2050, will be the country with the highest proportion of elderly people. Well, really, this is very worrisome because we have developed a society which thinks about young people and adults, but we've never thought that there would be so many elderly people. We're going to have elderly people with healthy lives, with very good living conditions, but who have nothing to do. And besides, there are new diseases which are associated to this advanced age. And this includes degenerative diseases, mainly neurodegenerative diseases. We know that Parkinson's disease is one of those diseases, Alzheimer's disease too, the two most frequent ones. But what is not true is that if we lived long enough, we would all end up having Parkinson's disease or Alzheimer's disease, or both. But I don't think it is. It is so. Parkinson's disease and Alzheimer's disease are two completely different diseases, illnesses. So the fact of having such a high number of degenerative diseases in elderly people and a very high prevalence, because we are working now with figures that indicate that 20 to 25% of the people older than 65 years are going to have a cognitive deterioration, which will be the anti-chamber of Alzheimer's disease. Now there is a good proportion of the population older than 65 having Alzheimer's disease. We may have 700,000 people with Alzheimer's disease. Most of them don't even know that, and they have not even been diagnosed as having Alzheimer's disease. What do we want to know about Alzheimer's disease? First of all, how the disease emerges, not why, but how. And then, if Cajal was alive, he would continue to persecute those butterflies, because we still don't know much about how the nervous system works or the neurons work. I think that that human biology is extremely complex. Things are not as simple as we would like them to be, in order to understand those processes. Since the times of that crippling's disciple, whose name was Alzheimer, who first described what was happening in a patient and described senile plaques in the brain with a protein which was procyloid and then neurofibrillar both. Well, not much has been discovered about those proteins. We don't know if they accumulate or why. And we know their nature. They are glucoproteins, and this involves a lot of complexity in the synthesis, in the transportation, and in this case, these are when they change. Well, these proteins are necessary, but we still don't know their physiological role. We just know that they are the cause of Alzheimer's disease. So we are still in very early stages. Most of the information we have about Alzheimer's disease has been obtained through transgenic mice, which mimic Alzheimer's disease, which is suffered by a certain proportion of the population. But we don't know what happens in most patients with Alzheimer and what happens with their genes. And in this regard, we still have a way to go, which is extraordinarily long, to know what happens in Alzheimer's disease. The obvious fact is that at present, the drugs we're using are drugs that come from the information that became available 20 or 25 years ago. There has been nothing new allowing us to develop new drugs against Alzheimer's disease. So we are facing a very important challenge, and we have a lot to work along those lines. Well, to the left of Dr. Regidor, we have Andrés Martínez, foundation of the founder of the foundation, a scientist who carries out his work in a difficult environment in developing countries. What should we do so that all medical research, the new medical and biomedical technology reaches those developing areas? Because the inequalities are outrageous. How can we succeed in sending those new technologies to us some extent? Well,
Sí, ¿se oye? Bien. Decía que preparando esta intervención,
as I was preparing this presentation, I was focusing on the title of the Campus of Excellence that mentioned the university people and business people with regards to the Millennium Objectives. And my foundation, the foundation where I work, is especially dedicated to that. In the areas where I work, there is not many cases of Parkinson's disease. There are very few cases of cocaine addicts, and people most of the time do not get dementia because they're young at a young age. If we consider the Millennium Objective, the 8 of them, with 18 targets, those related to health indicate that by 2015, compared to the year 2000, we should reduce children's mortality by 2/3. And the first two main causes of children's mortality are acute diarrhea and acute respiratory diseases or infections. Besides, they tell us that we should reduce maternal mortality by 2/4, and this is related to birth or delivery complications and pregnancy complications. And then another objective, another target is to try to revert one of the most serious diseases in very remote rural areas, namely malaria. The problem is how to reduce all this in areas, well, because most of this mortality takes place in areas where there are no roads, no electricity, no electrical light. The primary healthcare system is organized in centers where there are doctors and healthcare outposts which do not even have doctors or even nurses. There are technicians, health technicians. I would like to offer you an alternative in which we are working to try to support the development, the achievement of the Millennium Objectives. We mainly try to establish a communication between that isolated technician and the reference physician. So what we do is telecommunications for health. We try to improve healthcare through the design and the appropriate use of information and communication technologies. And we're working in 3 areas. First of all, the technological one. We said that there are no cable telephony networks, there is no cellular telephony. The only traditional communication is satellite communication, but the operation costs cannot be afforded in those areas. For years, we have been working in trying and achieving voice and data communication through radio systems. We can use emails through shortwave and VHF radio, and in the last few years we have modified the Wi-Fi protocol, which works in short distances up to 300 meters, and through small modifications we were able to have them operate in point-to-point distances of up to 100 kilometers. The latest one was mounted in the Napo River in Peru from the city of Iquitos up to the border with Ecuador. We set up a border network of 150 kilometers, which communicates up to 58 healthcare establishments, and all those where there is no physician are communicated with reference hospitals. We're using with technology, but this is just a tool. What do we eventually want to do with this technology? First of all, to improve the epidemiological system. Before, in the past, we had to travel to obtain those reports. Now they are sent via email. The information comes on time without mistakes. Secondly, improving the diagnostic capacity of those more isolated centers without a doctor, just by being able to consult when they have a doubt. Now it takes place through IP telephony. They raise the receiver and can ask questions to the reference doctor. But, for example, in the case of acute respiratory diseases, the reference doctor needs more information. He would require doing an auscultation of the patient. We have a system of wireless stethoscopy. So the a physician through a video conference sees where the other doctor places the stethoscope, and the results enter the computer, and through Skype, for example, these data are sent to the reference doctor, who will be able to see whether that is bronchitis or pneumonia. If it is bronchitis, he will indicate the medication. If it is nose, the patient will be transferred to them. The objective is to save urgent transfers, because in those areas little planes or boats are used, and well, these things are very scarce. The next objective is, if we do not succeed in avoiding this transfer, at least let's coordinate it better and let's take less time for doing that. Thank you again to the telecommunication systems, and we have been able to reduce by 60% the time of transfer of urgent patients. In a video conference that I had last Thursday with this network of Napoleon, Peru, the medical coordinator told me, are these systems well installed? We haven't had any mother disease. I'm going to talk about something completely different now. One of our efforts has consisted in changing the mentality of the universities in developing countries, who were focusing on the same thing as scientists in developed countries. And just if they turned their head around, they realized that they had real problems of their company that they themselves can solve better than anybody else. Other scientists here, I believe, would be willing to say that the first step for a big invention or innovation is to know the problem quite well. And this is the only advantage they had with regards to the scientists of the developing countries. Developed countries, sorry.
José Antonio López Guerrero from the University of Madrid. And now we're going to start with a very concise question: among risk factors for neurodegenerative diseases, might we envisage the possibility of viruses? Thank you. I'd like to say that for me it's a great honor and a pleasure to share this panel session with outstanding experts in the field, and I'd like to comment on the fact that as a full professor, scientist, and director of scientific activities at the University of Madrid, I am just sharing my time in the field of research in neurobiological sciences, and also as part of scientific dissemination programs. I think that is part of research. And first of all, prior to answering to your question, well, that's been previously answered, because we've said we do not know the etiological factors. I thought you were going to give us news. Yes, I know about the etiology of multiple sclerosis. This is a field I try to work on. Yes, it's been said that when Francis Crick, a few years ago, he said at the pub in England, saying, "I've discovered the molecule of life," then he got the Nobel Prize award for that. He thought he had concluded his research in the field of biological molecular biology, and he decided to change gears because he wanted to discover something else, perhaps looking for a second award, and he went to San Diego to study neurobiological sciences, thinking that next step would be as important as DNA and the way of brains functioning. He passed away 2 years ago, and he might have had another life to live, but it wouldn't be enough to discover that, because molecular mechanisms of communication, intracellular communication, even though it's been said before that there are communication through ion channels, neuronal communication, but the final mechanism inside the cell and in between cells, all cells. When we talk about neurons, we talk about neurons as if that type of cells were the only type. There are other cells in the brain, the glia. Glia, as you know, are as important or even more important than neurons because they protect and they are 10 times more frequently encountered than neurons. And communication between glia are oligodendrocytes and neurons at the central CNS or the swan cells at the peripheral system are far away from being totally ascertained. And the first step towards a discovery of something new is to ask the proper question, to formulate the proper question. And in the case of here neurodegenerative diseases, we've been talking about Parkinson's disease, Alzheimer's disease, and I had the opportunity of, as part of the group of Professor Valdivieso at the Molecular Biology Center, we were trying to work with transgenic animal models, trying to study Alzheimer's in murine models, and it's been mentioned here, oligodendrocyte degenerations that might trigger, to a certain extent, a disease that's a neurodegenerative disease, a chronic disease, like multiple sclerosis. How about the causes of multiple sclerosis, etiology of these diseases? It's been mentioned here. We have a small percentage of dementia, pre-senile dementia, familiar Alzheimer, and this is not the one we know, but this starts quite early in life, sooner and earlier than senile dementia. In the case of the elderly, it is clearly seen that punctual mutations at given genes are able to trigger events known, marks known, like amyloid plates or neuronal configurations like bundles, and then 90% of our elderly people, they have multiple factors, environmental health factors, working in the case of aluminum production or being a woman or trauma. Those were factors that we might know, and there were also other factors, genetic factors. It is known that there are genes implied in the production of a risk factor, the development of neurodegenerative processes like ApoE transporter of that's been statistically studied, and it's been also proposed a couple of weeks ago. There was a publication, several papers were published in Nature, and one of the most wonderful journals worldwide, with the screening, analyzing a given number of genes. They could localize a small number of genes implied in possibly the protection of brain damage or against brain damage. So when you alter those genes, you have a risk factor for developing Alzheimer's disease. It's been said that the genes represent 25% of the risk of developing Alzheimer's. Those are causal effects, 75% of unknown causes in the case of Alzheimer's disease. And a daring approach was proposed by a lady scientist, and Rudy from Manchester. She thinks she had discovered a very important risk factor, and I'm almost afraid to mention this because it is the herpes virus. Having herpes viridae viruses, well, it's something that's been implied in neurodegenerative processes. She thinks herpes might be related to Alzheimer's. We have here Alzheimer's disease experts, and I'm going to talk about multiple sclerosis also because our current research shows that members of the families of herpes, and we have 9 species that are inducing, for instance, herpes 1 or Epstein-Barr herpes or virus that's been implied. And we have genetic studies, statistical studies, and that is prone to controversies. And one of the viruses is the herpes simplex type 1. As Ruth says, having a virus all your life, 90% of the population of the brain, that is not a good thing. And our line is not saying this is the etiological cause, but we could have a virus as a risk factor. Why is it so? Well, that we'll have to determine in the case of multiple sclerosis, but at very many neurodegenerative processes, because we do not know initial causes, no etiological agents of diseases, basically neurodegenerative diseases, as well as we do not know agents of autoimmune diseases. Thank you. And here we have an expert in the case of cancer, Dr. Manuel Esteller from the Center for Oncological Research. Dr. Esteller, how about the news? What are you expecting to see published? Something you wait, your finger crossed, but being realistic, what do you expect? Yes, thank you. Today we had some news. If you read the newspaper, you will see that mortality due to cancer has been reduced, and since 1995, now we are at 2009, and those are good news. What we're talking about is if we could see there is a cure for cancer. Those are different news. We do not have penicillin against cancer. That is a multifactorial disease. At the beginning, people thought it was due to viruses. There were research centers in the States. They thought it was only due to viruses. Then people thought that's due to genetics. And now we say epigenetics, the regulation of genome. And of course, there are 3 concepts: pathological agents, pathogenic agents like viruses or bacteria. In the case of GI cancer, some genetic factors and some epigenetic factors, a combination of the 3 of those factors, and then some others. And I would say that, of course, we all know that alterations of ubiquitin are modifying the expression of genes, and that we see in human pathologies, and also the expression of genes regulating proteins that are histones. Histones are regulating the human genome. What we do in our field is to demonstrate that cancer is a polygenetic condition, but at the same time it's an epigenetic condition. Epigenetics will be the regulation of genome. We say genetics is like the hardware, epigenetics like the software we introduce here to explain everything, and we have examples like cloned animals. When you clone an animal, this DNA is identical, but the animal we've created is different because genes are the same, but the regulation of the genes is different because life leaves chemical signatures our genome that means we are the way we are, and we could have certain diseases. Another case, monozygotic twins. They have the same DNA. They should be identical. If they are not, it's because of a different epigenetics, chemical modifications, chemical situations like ADN methylation, methyl group here, histones. DNA is not a double strain. It's not like Watson Crick double chain, but we have a three-dimensional structure to fit it within the cell. DNA will have a 2-meter DNA, and you need, it's like a string of pearls formed of histones. If they are compressed, they wouldn't allow gene expression. Histones are altered, DNA methylation is altered in the case of cancer, and there are drugs that will revert processes. Only 4 epigenetic drugs approved by healthcare systems to treat lymphomas, and that the drugs are reprogramming cells that do not understand signaling arriving. It's like having an information when you have a virus, and then that's been approved for treating leukemia, lymphomas, epigenetics in cancer. That's going to be applied or applicable into different pathologies. It was published in Lancet that neurodegenerative diseases will have or show epigenetic alterations that are very important. As we do not know about the causes, perhaps this will be very important, epigenetically speaking. We do have drugs, so it is worthwhile to approve or test those drugs in murine models. So, to summarize, I'd like to say that viruses, and we're talking about viruses, many people are infected by viruses, but only a few are expressing the disease. Human papilloma, there are lots of youngsters infected, but this won't progress. Why? Because the virus is having epigenetical changes and they are regressing. The genome of the virus is not changing, but the regulation is changing in the case of healthy carriers and those expressing the disease. I'm summarizing partly in my introduction saying that in cancer we have polygenetic conditions. Those are also epigenetic conditions, environmental conditions, and this is a combination that makes them difficult to treat. Specifically, thank you. I do have here 2 Nobel Prize laureates and some other people, very wonderful people. Perhaps the best question is, or the best thing is to give the floor to the participants. You know, you can put in writing your questions, and you have those little cards. You can send it to all the specialists.
Anyone here will take the question and will answer, or either you could address the question ad hominem. And, well, now we can open a second round of interventions después de lo que hemos oído. Si queréis, cualquiera de vosotros, any one of you can...
any one of you...
world-renowned scientist asking questions. Please, Doctor.
Okay, well, let me just make a comment to what has been said about the degenerative diseases by Professor Guerrero.
Thanks a lot.
Regidor, from the standpoint of the basic sciences. And let me summarize it in 2 statements. There is some, I think, from my standpoint, some bad news, and there is some good news. The bad news is that, of course, understanding the brain is a very difficult thing to do. The 1990s were declared by the US Congress as a decade of the brain, with the hope that towards the end of this decade one would know more about the brain. It didn't happen. I think it needs maybe another century to really understand what happens in our brain. So it may be difficult to cure certain diseases, which are mental diseases. The good news, I think, is that particularly the diseases, the degenerative diseases, are happening on a cellular level. It may be enough to understand the cell biology of the neuron, which is much more circumscribed than the big problem of understanding the brain. Or it may be enough to understand the immunology of nerve cells in order to get or it may be enough to understand certain aspects of cell cycle regulation and protein degradation, you know, to to get a handle on degeneration. This is, and it's a good news in the sense that it's much more focused kind of problem than the big problem of understanding the brain.
Thanks a lot. Interesantísima precisión.
Interesting statement. And here to my left, to my right, please, Dr. Carballo.
A mí me gustaría seguir la...
I'd like to continue in the same line of thoughts what was said by Martínez and the challenges of maternal and child mortality. In the same line of thoughts, and I had participating at a debate, and they were putting the guilt on or the blame on pharmaceutical companies. But I think that when we talk about the developmental challenges, we have a challenge there because well-off countries feel guilty. It is good to acknowledge that most of the countries having high maternal mortality and child mortality are not very poor countries. They are just countries where they did not know how to use the resources they have. They have excellent schools like Ghana, Nigeria, Kenya, Tanzania, Uganda. Excellent medical schools they have. So I think that in this debate on how to cope with the challenges, what is required is not only investing in medical sciences or in healthcare fields, but in political efforts. I think that in these countries, and I'm going to be a bit, cynical, because at the highest level there is no political commitment, no political will for national investment in development of healthcare services for a country. That is, of course, there is a scarcity of resources, but those are countries where, unfortunately, they export lots of skilled medical doctors who are going into Europe, the United States, excellent doctors and nurses. Those headhunters invite them to go to other countries, and we should work and enhance political will more, and also at the same time as we develop international aid. Thank you. It's a very interesting contribution. And now, while the doctor is making a statement, I'm going to take questions for consultations. Yes, well, as it was said, those countries are not necessarily very poor countries, but there are differences. There are internal differences that are huge. And politically speaking, we should try to change that situation. I am a professor at Rey Juan Carlos University. At the foundation where I work, we have different universities, Spanish universities, Polytechnical University from Madrid, University King John Charles, Peruvian universities like the Catholic University from Peru, the Cauca University from Colombia. Regarding scientific fields, we try to find solutions to problems of the first world. It's very difficult to publish an article concerning a problem that's restricted to developing countries, where results are applicable only to those countries and not applicable to developed countries. Those professors from these universities from the south, they are focused on finding the same solutions as we are looking for. If they do not have the same problems, they do not get the same solutions. So what I'm saying here, what I request in the new legal framework in Spain regarding scientific research, we have an item where we scientists we're going to be able to look for solutions that are not applicable in Europe, but only applicable in developing countries. For the first time, there was good coordination, the Ministry of Foreign Affairs and the Ministry of Science and Technology. And I think it is a very good opportunity to implement this. Please try to be very brief, because we have questions from the participants. We're just waiting for the scientific framework. And now that you're talking about financial resources, we should say that we've signed the manifesto against reducing the budget for R&D. The ministry, well, they are threatening us. They're masking the situation. The situation is not clear. But they're going to reduce, they're going to cut down resources. So they're not really trying to give fuel to the only engine that's able to take a country out of a crisis. That's what Dr. Gabilondo said. They are reducing funds allocated to research and development. We do not know about percentages. We don't know if those are structural funds or project funds, but who is going to give the fuel for progress is not realizing that even countries that are behind, like Greece and other countries, are investing more. They are reducing money from other parts, but to enhance research and development. If they don't understand this, with a reduction of a budget to try to get money for international aid, I think it's complicated. That's not my statement. That was said by the edition a couple of weeks ago, and I think our guests have read that article where it was stated. Yes, 2 minutes. Sorry. Doctor, yes.
About, well, this session is also called about future directions, so maybe we should end. And we talked about neurodegenerative diseases. So I think we can be optimistic about the future, maybe not in the next year or two, but in the future, because it's becoming clear that most of the major neurodegenerative diseases like Alzheimer's disease or Parkinson's disease are due to the accumulation of abnormal proteins in certain neurons.
At your field, sir?
Yeah, in the cell. Abnormal proteins are formed because they are damaged, so we are learning what are the damages. And once they are damaged, several things can happen. So some of the abnormal proteins became aggregates. Some of the proteins do not aggregate, but then maybe even they are more harmful because then they compete with normal proteins, do not let normal proteins to work. And we also have some abnormal proteins that become fibrous. Like amyloid fibrils, which definitely either in the cells or outside the cells, they can do damage. Now, if we understand more about why these are formed and how, and then we can prevent the formation. Or if we understand how to deal with this situation, abnormal proteins can be degraded by the ubiquitin system, so we can beef up the ubiquitin system. We can channel it towards these abnormal proteins. We can also try to refold these abnormal proteins because there are other proteins that are called molecular chaperones that help to refold abnormal proteins to the right way. So we can also manipulate the chaperone, the refolding machinery. So I think that in the long run, we can be optimistic about neurodegenerative diseases in the future, and it will take time, but I think eventually we'll get there. So that was my comment about.
Thanks. Very hopeful. Very hopeful. We need that. We need lots of hope. Por favor.
Yes, lots of hope. My question goes to Dr. Carballo. Doctor, could we really, universities, well, not considering all financial restrictions, I hope the legal framework for sciences, as a rector, I've been told many times about it and we're still waiting for that law, but I hope this will be passed someday Do you think that from universities where we are now having different interests in favor of international cooperation and development, do you think we can play a political role, or we should get together with someone else, I mean, with another one, to have a specific weight big enough? Well, I think that there are universities in Spain that should get together, work together, close work with universities, for instance, in Latin American countries and African countries, developing centers in search for excellence in those countries. With what I said before, I didn't want to give the impression that we do not need a certain level. I don't want to use the word help, but cooperation. We need to cooperate with countries, developing countries, and I think that the challenge for the future, and I am very optimistic here, the challenge is to elaborate joint programs with universities, African universities and Latin American universities. And well, what about problems in those countries? We'll have to identify those problems and to develop R&D programs adapted to real necessities, applied to the reality, not applied to our reality or what we do think reality is, something we've seen from here, from Europe. This is not, because those are not the real things most of the times. And I think that regarding maternal mortality, for instance, historically speaking, how did it go downwards, I mean, as a trend in Europe? It was not due to medical efforts, not due to medical doctors, but mortality rate went down due to nurses and knowing how to assist at labor. And they were not very well specialized, but that was a big investment that was done at the level of the countries with a political will to invest. And I think it is important, and in African countries this should be the case. And I think that universities should do this, should work together, and this is what happened in the case of those programs with midwives. And well, that's one example. We could have political will and healthcare policies applying knowledge we do have, but they also have knowledge. And midwives, for instance, in Latin American countries where I had worked a lot, those are well, those are people knowing well what to do, and what they need is resources, support at the level of, and it is very important. Midwives were helping, and this is very important for strengthening quality of services with a strong political will. Applied research programs, appropriated programs with an excellent support from the administration, financial support, I mean. And as I said, for me, the future of scientific development and healthcare services in general is a joint effort. Universities in Europe and universities to those countries, Latin American countries and African countries. I'm not talking about other parts of the world because it's already an ongoing project. But in Africa and Latin American countries, we have very many pending subjects, things we should do.
La recta final ya en este debate.
Last part of the debate, we have questions from the participants. The question goes to Andrés Martínez. Do you think it's feasible to use the same techniques? I suppose there are other questions. Same techniques which were mentioned in the case of developing countries at rural areas. We could apply those techniques that were used in developing countries in our rural areas in Spain. The concept of services service concept when we talk about remote medicine, remote ecography, or whatever, to reduce costs and to improve the use of specialists. Yes, but technically speaking, modifying Wi-Fi for long distances and having reduced cost networks, no, because Spanish legislation is very restrictive while using the power, and we cannot use that. We cannot get into long distances because of restrictions. Question here to Rosario Morataya. Cocaine is a drug. It's a social drug. The question to the lady doctor: could we treat the addiction of, cocaine? Could we treat it as a disease? And it's cocaine use. How could we handle that?
Well, I think that people are social by par excellence. So, if you remove society from individuals, they stop being individuals. Yes, if we can remove sociability from them, perhaps we would be able to reduce drug addiction, more specifically to cocaine. But anyway, once the individual has become an addict, I don't think this has a very big influence on his or her addiction, because he is personally preconditioned in the central nervous system, which obliges him to go on taking that drug. And of course, there are characteristics in people's personality that makes them more vulnerable to the use of cocaine. And recently, the Science magazine, there was a publication by the group of Barry Everett from England, in that there were some traits in people's personality, for example, impulsiveness. They realized that in rats with a high level of impulsiveness, they also had a high rate of drug consumption, self-administration. In those rats, it was easier for them to self-administer cocaine. To some extent, society does have an influence, but once that the barrier of the voluntary use has been removed, that is, to the compulsive use of any drug, cocaine, with regards to cocaine, I don't think that society would have such a big impact on stopping the addiction. Even more, the individual would require to be determined to quit for personal reasons or family reasons, etc. And besides, he should have pharmacological help. For cocaine addicts, it is especially difficult because the limbic system, as soon as those persons pass a place where they had drugs, this provokes a very high rate of relapse. It's just passing that place. This is not the same with other addictions such as heroin or morphine. Once physical abstinence has been achieved, it's easier, or has finished, it is not so easy to relapse. Thank you very much. Then we will finish the panel. I'm just going to ask you to give me 2 more minutes for a final message that may help us go back home more reconciled with human and medical issues. A question to Dr. Carballo, just a couple of minutes. Why do you think that the purchasing process of retrovirals against HIV was characterized by slowness? This is a provoking question. I don't think it was slow. I think that it was highly accelerated through a period of about 8 years. This is when those retrovirals were obtained. I think that the field of AIDS thanks to political support and due to the interest of the stakeholders, has witnessed a tremendous progress. And this has not been seen, for example, in the area of tuberculosis, hepatitis, diseases which have been almost forgotten today. So I don't think this was a slow process, but just the opposite. You have been very clear. The last question is not addressed to anybody in particular. Which is the progress that has been made in chemical mental diseases such as bipolar disorders, schizophrenia, etc.? The answer comes now from heaven. Well, go ahead. I'm not a specialist in this area, but I'm just worried for what happens in the brain. What I do believe is that every mental disease, any disease which has the nervous system as a substrate, in this case the brain, I wouldn't call it a chemical disease, because the brain is not just chemistry. It is a physical, a chemical and biological compound, so to say. But major progress is being made in knowing the substrates behind those diseases. And this is very important because the treatments are becoming more and more specific with less side effects, etc. But it doesn't mean that, like in neurodegenerative diseases, we are far from healing them. And if the chairman allows me, I will abuse my 2 final minutes. In all those diseases, Alzheimer's disease, Parkinson's disease, multiple sclerosis, or even AIDS, very often we pay the necessary attention to the disease. But, well, however, we don't pay any attention to the patient, to the sufferer. And at present, this is very important, especially diseases such as Alzheimer's disease, in which the patient loses one of the most important abilities of human beings, that is being able to communicate with words. In this regard, we make a very big error to try to replace those needs with medication, which is completely inappropriate and inadequate, which only aggravates the disease or the situation of the patient. So I make a plea. When we are in front of a disease, we must work with it scientifically, but we should never forget that in most cases, behind there is a patient who is fragile and helpless, and we should do whatever we can to be with them, to work with them, and to improve their life quality and their dignity as a person. Fine, Dr. Guerrero. We're going to proceed the other way around. Just a couple of minutes to finish off. Well, Carl Wagner, a biologist, I think he was the one who said that the receptors of a virus in a cell were proportional to the number of dollars that were invested in research. Therefore, if we want to discover really the origin of diseases, because until we do so and find the molecular process of any neurodegenerative disease or even immune diseases such as diabetes, we can only give palliative treatments, which are important, but cannot be final. They cannot put an end to the pathological process. So my message is just a plea to invest more in research in Spain, because even at times of crisis, if something can take us out of the crisis, are two things: research and social dissemination. Thank you very much. I'm going to be very brief, and I'm always traveling between developing and developed countries. I would like to see in my field, which is medicine, to see the disappearance of this paradox, to have a solution in search of a problem in a developing country, but rather I wish we could cooperate in knowledge, the knowledge of the problems and the search for solutions. Doctor, well, I would like to finish with on a positive note. Really, although the studies on this vaccine against cocaine addiction have been disappointing, I would also like to say to you that there are other studies in clinical phase and phase 2 clinically with results which are encouraging, but they have not published yet, with acetylcysteine, a molecule which is used against COPD, chronic obstructive pulmonary disease. And this has provided spectacular results in animals, not only because they block cocaine administration, but also because it reverts all the plastic changes that occur in the brain. And acetylcysteine doesn't require any more introduction. It's a mucolytic agent. Yes, and when you take it, you don't feel like taking cocaine or having cocaine, but these are the results of phase 2 clinical studies that have not published yet. All the results obtained in animal trials are spectacular, and they have been published in Science, Neuroscience, Nature, Neuro, in many journals. They, by Peter Calibas mainly, which is a group in South Carolina University in Charleston. These are good news. Dr. Hersko.
I'm sure, I'm not sure I followed everything in Spanish. So what is the question now?
No, we expect you. Sorry, I didn't. I thought that they were translating.
No?
I will repeat for you. No problem. We expect you to say a nice farewell with if you want, as much positive as you want on medicine and mankind, and so we can go home with a smile in our faces.
With a smile, yeah. Well, if you look back, maybe we can look at the future from looking back. If we look back 30 years from today, the medicine has progressed greatly, huge progress. Well, you can see it by life expectancy that went up from a little bit above 70 to 80. That's average, so most of us are better than average, I hope. And that is because there were huge strides in medicine. There is a lot about the prevention of disease. That's most important. So the realization of the importance of better blood pressure medications, better cholesterol medications, maybe lifestyle changes. Just stopping smoking, doing little exercise, and trying to keep fit. That makes a huge difference. And maybe the biggest revolution in medicine was technological revolution. That is 30 years from ago, we didn't have computerized tomography, we didn't have MRI, we didn't have all the imaging that makes such a big change in medical diagnosis, medical treatment. So if we'll project from the next, and that is all due to research. And by research, I mean basic research. All the applied research was based on basic research. So this is all eventually due to research, which was based on basic research. So if we can project from there to the future, so I think that life expectancy will go up even more. And society will have to cope with it. It will be able to cope with it because not only the length, but also the quality of life, I think, will be better. So we can retire later and we can work longer. So that's good. I think that we will have new medicines, as I just mentioned to you. Our basic research that now can help certain patients with cancer. So we'll have better medicines also. So on the whole, I think that that medical future is quite good. I hope that societies will be wise enough to cope with the huge progress that we'll have in medicine.
Good words, thank you. Dr. Carballo.
Sí. Mira, genéticamente yo soy...
Genetically speaking, I am optimistic, and no doubt we are at a century that is full of, and will be full of, extraordinary biomedical advances. No doubts about it. And in the same line of thoughts, at the same time, we'll have to look for ways of improving our relationships with patients. 60% of all people of a given age, and I'm not going to use the word old people, because whenever you were talking about old people, you were looking at me, staring at me, but 60% of elderly people come into our office when they are 60 plus. Those are not visits due to medical problems. They come to see us because of psychological trouble, psychosocial problems, and they are in search for support, having a debate, clarifying things, etc. And this is what we are going to take into consideration at the same time when we invest what we're going to invest without any doubts. In the field of biomedical sciences, we'll have to get a better relationship with our clients, with our patients. Thank you very much. Doctor, you have the floor, sir.
My comment will be very similar to that of Dr. Hersko, but let me start from a different point. Most of the so-called incurable diseases, I think, are diseases of the brain. And they are so because the brain is a terribly complex organ. And as 100 years after Cajal, we know a lot more about the brain, that's for sure. And as I pointed out, there is hope for progress on certain classes of diseases, the degenerative diseases, for instance. Other diseases like the mental diseases, schizophrenia, bipolar disorders, they may need a more complete understanding of the brain. But even that, I think, will eventually come. And as I said, 100 years after Cajal, we know a lot more. In another 100 years, we may be able to have all of this under control.
That's good to know. Doctor Esteller, llegó el último y se va el último.
Now, last but not least,
So in oncology, in the 1940s, survival was only 5–10% at 5 years. Today, survival at 5 years is 65%, total cancers. Their cancers will do much better. Breast cancer, 95% survival, 5 years. Others, very little, glioma, pancreas, only 1 year survival. So we need a lot of effort there. And this change from survival 10% to survival 60% is not random chance. This is research. Applied research, basic research, all this research. And this is the reason for this advance. And this research sometimes is expensive, it requires money. This money came sometimes from the public part, but also has to come from charities. And for example, in Spain, this is completely underdeveloped. We don't have charities giving a lot of money to research, and this will have a healthy envy from the UK and the States and other countries in this regard. The second question is, I'm going to give you a receipt for curing 30% of cancer. 30% is stop smoking. This is able to eliminate 30% of cancer. Lung cancer, bladder cancer, head and neck cancer. And this in numbers is 30% of the cancer in the world. Total numbers. And also a big problem in the third world now. A problem that is growing. A lot of smoking without any control. Finally, the final message is a challenge ahead is that people ask me, okay, now you're curing 60% of the cancer, 65%. What are you going to do next? Are you going to run out of your job? No, because there's a big challenge of the brain, really, like Rick said. So we know very little about the biology of the brain, extremely little, and biology even of the neurons and of the glia cells. So I think that there's a lot of work for all of us in the next years.
Bueno, ha sido la consulta médica más interesante que he oído muchos años.
The most interesting debates I've witnessed along very many years. And please, someone would be so kind as to tell us if they're getting the translation into English, because one of the speakers told it was not available.